brain you were born with is not the brain you are dying with. Between those two moments, there is a continuous, living, extraordinarily dynamic process of construction and deconstruction, of building and unbuilding, that neuroscientists have given the beautiful and precise name of neuroplasticity. For most of the 20th century, science operated under a dogma so limiting, so profoundly mistaken, that it constrained our entire understanding of human potential. The dogma was this: the adult brain is fixed, its structure is permanent, its circuits are sealed. You cannot teach an old dog new tricks, and you cannot grow new neurons once you have passed through childhood. This was wrong. Magnificently, liberatingly, irreversibly wrong. Long pause. The revolution began in earnest in the late 20th century with the work of researchers like Michael Merzenich, whose meticulous experiments with the somatosensory cortex of primates demonstrated is now one of the foundational principles of modern neuroscience. The cortical map, the brain's internal representation of the body, is not fixed in adulthood. It changes in real time in response to experience. Regions of the brain that represent heavily used body parts expand. Regions representing neglected body parts contract. The brain literally reshapes itself around what it repeatedly experiences and practices. Let us go deeper into the mechanism. Let us travel into a single sepse, into the microscopic space between one neuron and the next, and watch the miracle of neuroplasticity unfold at the molecular level. Deep breather. Neuron is one of the most beautiful structures in the known universe. Its cell body, called the soma, is a universe of biochemical activity, containing the cell's nucleus with its DNA, its mitochondria generating energy, its ribosomes manufacturing protein. From the soma extend to types of projections, the dendrites, which branch like winter trees in multiple directions, reaching out to receive signals from neighboring neurons, and the axon, a long, thin myelin-sheathed cable that conducts electrical signals away from the cell body and toward its target. The axon terminates in a structure of extraordinary elegance called the axon terminal, which does not quite touch the dendrite of the next neuron. Between them exists a gap of approximately 20 to 40 nanometers, almost incomprehensibly small, but enormously important. This gap is the sepse. It is the place where one neuron speaks to another. It is the place where the miracle of learning, of change, of growth, of reprogramming physically occurs. Neuroscience note: when an action potential reaches the axon terminal, it triggers the release of neurotransmitter molecules from vesicles into the synaptic cleft. These molecules diffuse across the gap and bind to receptor proteins on the postsynaptic membrane of the receiving neuron, either exciting it as in the case of glutamate or inhibiting it as in the case of GABA. The strength of this synaptic connection, how efficiently the signal is transmitted, is not fixed. It is modifiable through a process called synaptic plasticity, long-paused process by which SAPS is strengthened through repeated activation. The molecular basis of learning and memory is called long-term potentiation, or LTP. When two neurons consistently fire together, the SAPS between them undergoes physical changes. Receptor proteins are inserted into the postsynaptic membrane, making it more sensitive to incoming signals. The axon terminal becomes more efficient at releasing neurotransmitters. The connection becomes more robust, more reliable, more automatic. In a very real and measurable sense, a repeated thought, a repeated emotion, a repeated behavior changes the physical structure of the brain, and the reverse is also true. When a synaptic connection is not used, when a thought is not thought, when a behavioral pattern is not enacted, when a belief is not repeatedly reinforced, that connection weakens. Postsynaptic receptors are removed. The axon terminal becomes less efficient. Eventually, in a process called synaptic pruning, the connection may be dismantled entirely by the brain's cleanup crew, the glial cells. Deep breath. Let us speak about the glial cells for a moment, because they are among the most underappreciated heroes of the brain story. For decades, they were considered mere supporting actors, the scaffolding that held the real neurons in place. We now know this was a profound misunderstanding. Glial cells, particularly the astrocytes and microglia, are active, dynamic, essential participants in the brain's plasticity. Astrocytes, the star-shaped glial cells, physically enwrap synaptic connections. They regulate the chemical environment of the synapse, recycling neurotransmitters, neurons, literally shaping the conversation between neurons. And the microglia, the brain's immune cells, perform the pruning function, reaching out their tiny arms to engulf and consume synaptic connections that are no longer being used. They are the brain's editors, its curators, its gentle demolition crew, clearing away the old to make space for the new long possible. Perhaps the most magnificent discovery in the field of neuroplasticity is the phenomenon of neurogenesis, the birth of brand new neurons in the adult brain. For decades, this was considered impossible. The adult brain, it was believed, had all the neurons it would ever have, and the arrow pointed only one direction, toward loss. Then came the work of researchers like Elizabeth Gould and Fernando Nottebohm, and the paradigm shattered. New neurons are born primarily in the hippocampus and the olfactory bulb, continuously throughout adult life, and those new neurons are particularly sensitive to being incorporated into active neural circuits, meaning that what you think about, practice, and repeatedly experience determines whether these new neurons survive and are integrated, or whether they are pruned away within weeks of their birth. A key molecular player in this process is a protein called brain-derived neurotrophic factor, or BDNF, often called Miracle-Gro for the brain. BDNF is produced in response to physical exercise, to novel learning, to meaningful social connection, and critically to deep, restorative sleep. It promotes the survival of new neurons, encourages the growth of dendritic branches, and enhances synaptic plasticity throughout the brain. You are, right now, as you rest, creating the conditions for BDNF release. You are, right now, fertilizing the soil in which new neural pathways will grow. Deep breath, feel that. Let it land in your body, not just your mind. The brain you will wake up with tomorrow morning will be immeasurable in meaningful ways, different from the brain you went to sleep with tonight. New synaptic connections are being formed. Old ones are being evaluated and selectively pruned. New neurons born in your hippocampus are seeking circuits to join. And the thoughts, the images, the suggestions, the new stories that you allow into your mind in this theta state, these are the patterns around which those new circuits will form. You are not passive in this process. Even in sleep, even in the surrender of consciousness, your intention matters. The last thoughts you carry into the darkness are the scaffolding around which your sleeping brain builds its new architecture. This is not mysticism. It is neuroscience. It is the foundation upon which the rest of this evening is built. Deep breath, let us stay for a moment longer inside the neuron, because there is more beauty here than we have yet described. And beauty matters in the subconscious landscape. It matters because the subconscious responds to things that are vivid, felt, and resonant, and there is nothing in biology more vivid or miraculously resonant than the electrical life of a single human neuron. When a neuron receives enough excitatory input from its many synaptic connections, when enough, its neighbors say fire at the same moment, it reaches what is called the action potential threshold. At that moment, something extraordinary occurs. Sodium channels embedded in the neuron's membrane snap open, and sodium ions, positively charged particles that were waiting in high concentration outside the cell, flood inward with dramatic speed. The electrical charge across the membrane reverses from negative to positive in milliseconds. This reversal, this sudden electrical bloom, then propagates down the axon like a wave, like a spark traveling along a fuse, like a thought becoming real, long pausient. The speed of this propagation depends enormously on whether the axon is wrapped in myelin, that extraordinary white fatty sheath produced by the oligodendrocytes, the glial cells of the central nervous system. In unmyelinated axons, the signal travels at about 1 meter per second. In myelinated axons, the neural superhighways of the brain, it travels at up to 120 meters per second. This is why expertise feels effortless, why the concert pianist's fingers seem to move before thought, why the experienced surgeon's hands know what to do before the conscious mind has formulated the instruction. The myelination of frequently used neural pathways, which occurs through a process called activity-dependent myelination, driven by the repeated firing of those pathways, literally increases the speed of thought in those domains, and the speed of this propagation depends enormously on whether the axon is wrapped in myelin, that extraordinary white fatty sheath produced by the oligodendrocytes, the glial cells of the central nervous system. In unmyelinated axons, the signal travels at about 1 meter per second. In myelinated axons, the neural superhighways of the brain, it travels at up to 120 meters per second. This is why expertise feels effortless, why the concert pianist's fingers seem to move before thought, why the experienced surgeon's hands know what to do before the conscious mind has formulated the instruction. The myelination of frequently used neural pathways, which occurs through a process called activity-dependent myelination, driven by the repeated firing of those pathways, literally increases the speed of thought in those domains. And here is the extraordinary implication of this for the practice of subconscious reprogramming. When you repeatedly think a new thought, feel a new feeling, practice a new pattern of response, you are not merely creating new synaptic connections. You are initiating the process of myelination around those new pathways. You are building the neural superhighways of a new self. And over time, weeks, months, years of consistent practice, those new highways become as fast, as automatic, as effortlessly accessible as the old pathways of habit and conditioning.